So let's start with the today topic. It's about drug metabolisms. As you know, the drug biotransformation, the drug metabolism. Already, I have some classes. I already take some classes. So let's start. Uh, about the drug metabolisms, the drug biotransformation. Uh, you can see it's also called drug metabolism or drug biotransformation. Today, and about the most of the drug whenever you are taking medications for certain therapeutic purposes. So it must be eliminated. It must be eliminated from your body. So, uh, you know, uh, most most often eliminated by a biotransformation, and then are they excreted in the urine, bile. And there's a process of metabolisms. They mostly convert the more lipophilic drugs into more excretable products or more water soluble products. You can see from where is the liver is the major site for the drug metabolisms, but specific drugs they may undergo part of some other tissues like kidneys and intestines. They can also perform metabolize the drug products. And the basic already we have been through these slides, these lectures that I already discussed that in a little detail. That you can see the drug that enter. They have the metabolic process have different phase one, phase two reactions, and you can see the drug may enter into the phase one, where mostly the enzymatic degradations may occur, and then the phase two reactions may occur, where the drug, where mostly the conjugation reactions may proceed, may occur. And whenever the drug may become active drug from the phase one, some certain like functionalization may occur, uh, and along with that, in the phase two, mostly the conjugation reactions are involved, right? And whenever the drug passes through the various metabolic, it becomes inactive excretion. But sometimes, like some of the drug during the metabolism, the toxic intermediates may form, and these toxic intermediates may have some toxic effects on the body. And whenever it is inactivated to certain excision product, they may be excreted either in the urine or in the bile. So this is just like a little basic concept. And most of the drug you can see whenever the drug enter into the body, it must be eliminated. As you know, already we have discussed that some of the you can see just check my okay. Whenever the drug enter into the body, it must be eliminated. As you know, from the already we know about the drug absorption from the site of administration. It goes to the blood systemic circulation and then distributed to the whole body, and then the elimination process starts. And the elimination, which are the main, like the hepatic metabolisms, then the elimination in bile, then elimination in urine, and all these things, they have a very like they can decrease the plasma drug concentration exponentially, like you can see from here. And these elimination of the drug metabolisms may either occurs through the first order linear kinetics or the zero order nonlinear kinetics. And you know that how the linear and how you can differentiate between the first order and the zero order kinetics. Whenever, like you can see, it's a low dose, the drug metabolisms is the first order kinetic. Whenever the doses is increased, then the drug metabolism will follow the zero order kinetics. And you know about the differences between these two. Whenever, and these nonlinear kinetics, you can see this is the main differences here. like mostly the toxicities may be associated whenever the drug concentration increases in the plasma. Similarly, and the first order kinetics, the derivation, this is the equation that can be used for the rate of metabolisms, and this is the equation that can be used for the first order kinetics, and this is called Michaelis-Menten kinetics. This can be derived through proper there is a proper derivation. We can discuss that some other times. And zero order kinetics, you can see it's mostly the dependent on the amount of drug. And here the concentration depend upon the concentration of drug. So you can see this can be used for the zero order kinetics in order to derive to end up calculate the rate of drug metabolisms. And the reaction of metabolism mostly is it cannot efficiently eliminate excrete the lipophilic drugs. And really that can really crosses the cell membrane. So they can be reabsorbed in the distal tubules. So lipid soluble drug must first metabolize in the liver and using the general two sets of reactions. We call it phase 1 and phase 2 reactions. You can see from here the phase 1 reactions. This is also termed as enzymatic. The enzyme dependent phase metabolic reactions. While here in the phase 2, this is detoxification pathways. This is mostly the drug detoxifies, then it become inactivate. And enzymatic reactions, we'll discuss that later on, that which kind of enzymes are involved in this phase 1 reactions, and what are their major functions, and what are their major roles. So, and along with all that, phase 1 and phase 2 reactions, sometimes active metabolites, as I mentioned, that maybe sometimes active metabolites may form, and these may be appear in the form of toxicities or metabolic metabolites, toxic metabolites, and may cause toxicities. Now you can see from here, from this slide, whenever the drug that maybe passes into the phase 1 reactions, the drug may be inactivated, unchanged, or most often inactivated through these oxidation, reduction, and or hydrolysis, right? So in the phase 2 reaction, some drugs then directly, this is the conjugation products may be formed. This is the functional. Here the functionalization may occur, while here the conjugation reactions may occur, and the conjugated products may be easily excreted or inactive. That can be inactive and can be easily excreted from the body. From this slide, let's more elaborate this with this example. Like the drug molecules may enter into the phase during the phase 1. The reactive metabolites may form, and these reactive metabolites then enter into the phase 2 reaction, where the conjugations may occur. And these conjugations, they may be excreted, and either through the kidney in urines, or they may be also excreted in the bile. It can be excreted through the GIT tract. And the conjugation, sometimes it may be reuptake. The conjugation may be reuptake, and the cycle may be repeated for the conjugation reuptake. So that's why they are again metabolized by the like converted to conjugate and then excreted either to the kidneys or to the intestines. And you can see phase 1 reactions mostly involve the oxidation, reduction, or hydrolysis of the parent drug, and you can see they may be resulting in the conversion of more polar molecules. Where the phase 2 reactions, they involve the conjugation by coupling the drug or its metabolite to another molecule, such as the sclerochronic acid attachment, irisylation, or sulfate or glycines may be attached. And you can see the phase 1 reactions, like you can, let me just like phase 1 reaction mostly convert the lipophilic molecule to more polar molecule by the attachment or unmasking the polar function groups, such as hydroxyl or amino groups. And either the phase 1, either it may increase, decrease, or may leave unaltered the drug pharmacological activity. Remember this point. And hydrolytic cleavage is along with oxidation, reduction, chylation, dealkylation constitute phase 1 reaction of drug metabolisms. And it make ready the molecule for the phase 2 reactions. All metabolic alter the molecule chemically. While in the phase 2 reactions, you can see This is conjugation reaction, consists of the conjugation reactions from the phase one, and sufficiently they become make it more polar so that can easily be excreted by the kidneys. However, in a phase one metabolite, they are more lipophilic to be retained in the kidney tubules. So again, conjugation reaction that these xenobiotics are the exogenous substances such as glucuronic acid, uric acid, acetic amino acid results. These are the conjugate substances that may be attached to form conjugation reactions and make it more polar. So usually water-soluble compound then can be easily excreted and they become therapeutically inactive substances. So that's why they can be easily excreted from the kidneys. Remember that whenever glucuronic acid glucuronidation is the most common and the most important conjugation reaction, especially in neonates. Sometimes they have deficiency of this conjugating system, like the glucose 6-phosphate dehydrogenase deficiency. And the patient, the neonates, or the children that have deficiency with these, so they make the particularly vulnerable to certain kinds of drug like chloramphenicol. Which is mostly this chloramphenicol is an antibiotic, which is mostly inactivated by the addition of glucuronic acid. So the patients and neonates that have deficiencies, they may be vulnerable to the chloramphenicol toxicities, like some kind of toxicity associated with chloramphenicol. And this point should also be noted that sometimes drug possessing the hydroxyl the amino group or the carboxylic group may enter direct into phase two reactions. There's no like that must possess already they have the functional group that possessing these functional groups. So it directly enter into the phase two reaction where the conjugated with the phase one metabolism. And the high polar conjugates may be then extracted by the kidney or the bile, as I mentioned already. So let's like try to understand this one. This is your drug. If oxidation occurs, then the most lipophilic molecules may be formed. This is called electrophiles. You can see it here from here. Whenever the phase two reaction or hydrolysis or reduction of these compounds make them become more nucleophiles. This will become more electrophiles, and these are more. You can see this is the more lipophilic. While whenever electrophiles, so again the glutathione conjugations may occur. Mostly this happens with the electrophiles, and then may form it, make it more hydrophilic or inactivate by the attachment of glutathione conjugation. Well, in phase two, this occurs mostly in the phase two reaction. Remember this. What happened to the nucleophiles? Nucleophile mostly in the phase two reaction, the conjugation reaction, sulfation may occur, the acetylation may occur, or the glucuronic acid attachment may occur, and these may form these kinds of biconjugates, and they are more hydrophilic or more inactive substances, can easily be excreted from the body. Just remember this example again, like or a metabolism scheme. Phase one reaction. Look at this one. Is the phase one and is the phase two reactions. What happens to this drug molecule? So hydroxyl group, you can see the functionalization occur here, and a phase two reaction when it enters. So this is the conjugation. The attachment of this is the conjugation reactions may occur, and this may form either the attachment of these conjugate byproducts, and they make it more like inactivate this product, this active drug become inactivate here and maybe executed, and also make it more water soluble. This is the active drug of acetaminophen. You know the famous acetaminophen, very commonly used. So 96% chances they may be inactivated by this glucuronic acid glucuronidation, and the conjugate metabolites may be formed, so they become inactive to this pathway. But sometimes, like the cytochrome is another phase one, cytochrome P450 systems. We will discuss that later on. 4% chances may be. This is the substrate, also a good substrate for the cytochrome P450 systems, and that may form this benzoquinone amine, and this is a hepatotoxic. The intermediate substance may form, and this is hepatotoxic substances, and they may have the liver. They may be associated with the liver toxicities, and that's why acetaminophen have a liver toxicities, like in high doses. And again, it can be also converted by the glutathione into the conjugate metabolite, and then may become inactivated by the glutathione conjugation reactions, and it can be inactivated by this. Similarly, you can see this is the whole process of the metabolic process, the anterior hepatic cycles, and let me just explain this one. The drug entered to the stomach. So the drug may enter into the stomach. The biliary eliminations you can see from here. Again, the drug may be absorbed into the liver for the conjugation, and the phase 1 and phase 2 reactions may occur. So whenever they metabolize these things, so they can easily be the lipopedic. The yellow indicate the lipopedic drugs, while the blue indicate the hydropedic conjugation products. So if it's a hydropedic conjugation product, can easily be excreted via here, right? While if it is like the more lipopedic, it can be absorbed by the hepatic circulations and come to the liver for the conjugation to make it more hydropedic conjugating product, so it can be by the kidneys. So here you can see the phase 1. As I mentioned, this is a functionalization reaction. These are the main the metabolic reactions: oxidative, reductive, and hydrolytic, with the help of certain enzyme catalysis reactions. We will discuss that in our next slides. And they most introduce the polar. Like you can remember that polar function groups may be attached, these kinds. When in phase 2 reactions, these are the conjugation reaction you can see from here. This is the combination reactions, that we can say conjugation reactions, and mostly the polar and ionizable endogenous groups, so to achieve complete solubility, like the glucuronic acid attachment, the sulfuric acid, the dilation, etc. may be attached, a glutathione, etc. As I mentioned in my lectures, and can eliminate lower intermediate biological activities of most of the compounds. And this is the enzyme, and sometimes they're using the enzymatic phase 1 and phase 2, especially the phase 1 reactions. Mostly they're using the enzymes, like the phase 1 oxygenases enzyme are used, transferases or other enzymes can also be used. But along with that, the Cytochrome P450 system display a major role in the biotransformations. But there are certain kinds of enzymes that are not P450 system and Cytochrome P450 enzymes. We will discuss that later on, but they have a role in the biotransformation of the phase 1 reactions. And these are the examples: what kind of carbon and oxygen are dealkylation, other sulfur, sulfate, and deoxidation hydrolysis of these things. And these are the additives, sulfates, sometimes sulfur transferases. The phase 2 reactions are also initiated by the help of enzymes. Without enzymes, it's very most of the reaction and most of the biotransformation and most of all these can need some certain kind of enzymes. So I'm not going into detail of that. We will discuss that in our Cytochrome P450 system. But let us start the Cytochrome P450 system. So in some of my lecture, I mentioned that phase 1 reaction mostly they have mostly they're dependent on the Cytochrome P450 systems. They also called microsomal mixed function oxidases enzyme. That's also known as these kinds. A phase 1 reaction, those reactions that are utilizing the P450 systems. And you can see the drug like oxidation and reduction reactions may occur, like the involved drug metabolisms. They catalyze the drug modified NADP. You can see the drug modified with the help of these oxidized Cytochrome P450 systems, and then actually introduced through a reductive steps. This is the reductive steps example. This oxidation reduction reactions may occur, and this is the P450 systems can be coupled to NADPH, this one, with the help of cytochrome P450 oxyreductase. Let's elaborate it more efficiently so that you can know. Remember that this is a P450 system is an important metabolism, important for the metabolism of many endogenous compounds like steroids, lipids, etc. And for the exogenous substances are also termed as xenobiotics. Remember this term, xenobiotics, that it can be any foreign substances that are not produced inside the human body or inside the organisms. So all those substances that may be introduced, or they may be administered from the outside, but it may be synthesized from outside. So they are termed as xenobiotics. It may include drugs. It may include food additives, maybe certain pollutants. They can also be xenobiotics. Environmental substances may also be xenobiotics. They have their other effects. Now, how you will recognize the cytochrome P450 system? Remember that the class is, so there must be like CYP. So this will be the class, the classification, the nomenclature. Generally, they can be designated as CYP. This is the class. While subclass can be, you can see the subclass. You can write down the subclass like attachment of certain Arabic numbers or certain numbers. As cytochrome P, you can say like 3A. Let's suppose. So they become the subclass. While then, if this become the specific isoenzyme, so you must attach certain other factor like 4 or 3 or E or certain other numbers. So this become the isoenzyme, the specific isoenzymes. You know about isoenzymes. I'm not going to. They have the different isoenzymes can be defined. How you can define the isoenzyme is they have different amino acid sequences, but they catalyze the specific, catalyze the same chemical reaction. Remember that? You just recall your biochemistry knowledge, your chapter enzyme kinetic catalysis, that isoenzymes are those enzymes which have different amino acid sequences that catalyze the same chemical reactions. They are isoenzymes. So same is the case. These are the isoenzymes. You can say isozymes. You can also term as isozymes, and these can be and they can be found composed of many families of heme-containing isozymes that are located mostly found in the liver and a GIT tract. Remember, these are they are found in majority in these areas. The family, as I mentioned, so CYP is the class. Whenever the subclass is CYP3A, like CYP3A, and the specific isoenzymes can be you can see write down by adding certain number like CYP3A4. This is the specific isoenzymes. It represents a specific isoenzyme. The class can be represented with this one. So this is the general classification of these enzymes. And remember, there are many different genes and many different enzymes. There's the various 450 system known as isoforms. That's why they are formed. That because of their you can say let me just clear this one. Okay, so specificity. Now, as you know, the specific enzymes they have their substrates. Like they must be attached to certain specific drugs reagents, and there are like some have mentioned that there are more than 12 specific isozymes that can metabolize, that can catalyze various chemical reaction and metabolize. Most of the majority of the drugs are catalyzed by this 12 or more class of the cytochrome P450 system. But some have mentioned like six kinds of these P450 systems, cytochrome P450 systems that can catalyze most of the chemical reactions. And these are the percentage that are available that isozymes that majority of the drugs are catalyzed by these. Just remember these points. CYP3A4. I have a slide. I will show that to you guys so that you can better understand the percentage. Which cytochrome P450 system that so isomer, isozyme, isoform is used to catalyze certain kind of reactions. So I will just show you that one. So as I mentioned at amino acid, the genetic sequences, sometimes the genetic, the many genes, different enzyme, that's various P450 system with isoforms. These are called as isoforms. And the enzyme have the capacity to bind a large number of structurally diverse substances. This is not like maybe one drug have different isoforms can catalyze one drug, like for example, just like isoniazid, it can be catalyzed by various isoforms, not just like one specific isoforms. And before catalyzed reaction like these can be, let me show you some more detailed examples. You can see contained by cytochrome system isoforms. You can see the percentage. It's CYP3A4 by 5. 36% of the drug are catalyzed by cytochrome P450 systems. Then after that, this one comes the cytochrome P2D6, 19%. This one, then 11%. This one, 5. So you have to remember these cytochrome P450 systems, especially this one. They catalyze most of the drug metabolites. Okay, so representative drug metabolism by each of the isoform and human drug metabolism. 12 unique isoforms, as I mentioned. These isoforms have been identified as playing a major role in the human metabolisms, and other may be discovered. Like they also left some spaces for the new discoveries. And you can see from here. Let's just suppose this is the 12 isoforms, cytochrome P450 isoforms. And you can see the substrates. Aromatic hydrocaffeine, theophyllenes are mostly catalyzed by this one, right? By this isoform. Nicotine 5-fluorouracil. These are the drugs. These are the substrates. These are the substrate, or you can say these are the metabolites that can be catalyzed by these peglotoxins. They are catalyzed by this CYP2CA. Phenytoin, warfarin, non-steroidal drugs, that's mostly catalyzed by CYP2C9, right? And tricyclic codeine, dexamethasone, beta blocker, and antipsychotic are mostly catalyzed by this isoform of the cytochrome P450. And the polymorphisms, I will explain that later on, that what is polymorphism and what are the other. Okay, just remember this point. As CP3A7, this is unclear. It may be similar to CYP3A4. This one, maybe, and it present only in the fetus. Remember this. Not that the polymorphisms. Let's explain these points. So hope so you got this slide. So let's just explain the genetic variability of the P450 systems. Remember that P450 enzymatic exhibits a considerable genetic variability among various racial groups and individual groups. And mostly this variation in the genetic variability of the P450 systems, they may alter the drug efficacy as well as the adverse events may be associated with these kinds. For example, the CYP2D6, this is particular, they exhibit a genetic polymorphisms. Like if mutation occurs like CYP2D6 mutation results in a very low capacities, though it's occurred very low capacity to certain metabolites. For example, if individuals that obtain no benefit from the opioid analgesic codeine. If a person is deficient with this cytochrome P2D6 enzyme, they mostly activate. The drug is the codeine. This is not an active drug, and most activated whenever they are metabolized by this cytochrome P450 isoforms, they become active. So patients are people that have deficiency of this CYP2D6 isoform, so they are not benefited by the codeine analgesics. Similarly, you can also characterize the CYP2C secondary these isozymes, like the clopidogrel. You know, this is a very famous antiplatelet drugs or anticoagulant drugs, most used in cardiovascular diseases. So patient, they have there's this one, the patient who are CYP2Cs or poor metabolizers have a diminished antiplatelet effects when taking this drug, and an alternative medication should be considered. Though remember, this is clopidogrel is also a prodrug and the mostly actively activated by the cytochrome P450 this isoforms to convert that into active metabolite. As I already mentioned that maybe the prodrugs, certain drugs are prodrugs are those substances which whenever activate after metabolic, they activated after metabolism, so after proper metabolic reactions they may be activated. So now there are certain cytochrome P450 inducers. Sometimes it happens, right? Let me just explain these points. The cytochrome P450 dependent enzymes are an important target for pharmacokinetic interactions. Sometimes, for example, phenobarbital, rifampin, and carbamazepines are capable of inducing. They can produce cytochrome P450 isoenzyme. This results increased biotransformation of drugs. So if you are taking these phenobarbital, rifampin, carbamazepines, along with these drugs, you can see that from here. So they may induce some interactions. Drug pharmacokinetic interactions may occur. Either they may increase or decrease certain effects of one drug, or maybe decrease the effects of other drugs. So if you are like this one, they're capable of inducing this cytochrome CYP isozymes. So result can lead to significantly decrease in plasma concentration of drug metabolized by this CYP isozymes. And for example, it's a very general. This is a very general way. If these enzymes are produced, so it means that you are producing certain enzyme that can convert, that can rapidly metabolize these agents, these drugs, and they decrease the plasma drug concentration of these metabolites, and so they may lose the pharmacological effects. Let's suppose the rifampin, for example, rifampin is an anti-tubercular, anti-TB drugs. They significantly decrease the concentration of this protease inhibitors. The HIV, this is concentration of human deficiency virus protease enzymes, and mostly they're potentiated by the HIV drugs. So they can diminishing the ability to suppress HIV replication. So if you are like taking HIV drugs along with the rifampin, we have a class like rifampin, whenever we go to the rifampin, we will discuss this thing. So you can know better that this drug should be contraindicated along with the anti-tubercular drugs. And there's the, yes. Okay, so immunodeficiency viruses. So the rifampin is mostly contraindicated along with the HIV protease inhibitor drugs, right? The HIV drugs. Remember, and other thing. Okay, just let me. You can see like the various inducers that you can see from here. Sometimes non-fluoxetine, haloperidol, and this is the isoforms that can be induced. Remember this, induced. And next slide, I will discuss the inhibitors that which will inhibit certain kinds of cytochrome P450 systems. So these are the inhibitors. Sorry, these are the inducers, and these are the classic examples of these isozymes that are induced by this by the presence of these other drugs. So that's why that most of the drug interactions occur due to these kinds of interactions phenomena. What are the consequences of increased drug metabolisms? If like you are certain drugs induces certain cytochrome P450 systems, they increase the drug metabolism. So what are maybe the consequences? The first one is that it decreases the plasma drug concentration. As you know, if it decrease the plasma drug concentration, so it means the drug will be rapidly, as you know, the therapeutic range and the toxic range. So they're not going to the therapeutic range. If the concentration is decreased, it's not exceeding the maximum plasma concentration for therapeutic purposes. So that's why the drug is the plasma drug concentration is decreased, and they will have no therapeutic responses. Decrease the drug activity if metabolite is inactive, and increase the drug activity if metabolite is active. Decrease the therapeutic drug effects. The same as these things. In addition, sometimes, remember these points is very interesting. You can see the natural pollutants and the pollutants. They can also induce certain cytochrome P450 system, even the pollutant environments. They have their effects, like certain polyacyclic aromatic hydrocarbon. This is found in the air pollutants. They can induce cytochrome PIA isoform, and this has the implication on certain drug, for example, amitriptyline and warfarin. This is warfarin is also anticoagulant. Amitriptyline is antidepressant, and this is metabolized by this P450 systems. So the pollutants can induce this aromatic hydrocarbons, can induce the cytochrome PA, so they can decrease the therapeutic concentration of these agents. Remember that these pollutants and some natural substances may induce this P450 system, and that can alter. They can effects. They have the therapeutic concentration of especially the amitriptyline and the warfarin. If a patient is using that, so if you are in a pollutant environment, so you are taking some natural substances along with the herbal medicines, so they can distort the P450 systems. That's why most of the literature, they mention that you should not use the natural products along with certain medications, especially if you are taking certain antidepressants, or you are taking certain like these kinds of drugs. So they may enter, the interactions may occur, and they may either decrease or increase the therapeutic, the cytochrome P450. They may affect on this. Hope you guys got this point. So certain cytochrome P450 system inhibitors. So sometimes inhibition of drug metabolism can lead to a significant increase in plasma drug concentration. Sometimes they also can inhibit the cytochrome P450 systems. As we know, as I mentioned, that cytochrome P450 system are important for the drug biotransformations. If certain agents that inhibit this cytochrome P450 system, so there may be chances that maybe the drug is not converted into more, this A is not converted to more B. So what will happen? The concentration of this drug may be increased in the plasma. And if you are increasing this concentration, maybe they exceed the toxic, the therapeutic range, and maybe reach to the toxic range. And there they show maybe the toxicities may appear. Remember this. So this is the most common form of inhibition is through competition for the same isozyme. Some drugs, however, are capable of inhibiting reactions for which they are not substrate. For example, ketoconazole, this is an antifungal drug, leading to drug interactions. How? You can see numerous drug inhibit sort of dependent biotransfer pathways of warfarin. For example, omeprazole. If you are taking omeprazole along with the warfarin, as a potent inhibitor of these three cytochrome P4 isozyme involved in warfarin metabolism. So what will happen? If you omeprazole, they inhibit the cytochrome P4 isozymes, this one, and this is responsible for the metabolism of warfarin. So as a result, what will happen? The warfarin concentration will increase in the plasma, and they will have their toxicities. Warfarin is a neurotherapeutic index drugs. Remember this point also. When you are taking warfarin increase, which lead to better anticoagulant effect and increased risk of bleeding, this is the toxicities may appear, and in case of warfarin toxicities. This point should be noted. The more important cytochrome P450 inhibitors are erythromycins, ketoconazoles, and ritonavir, because they each inhibit several cytochrome P450 isoenzymes. And from here, let me just hope so it will be clear, little clear. Drug intake, absorption. So here from site of drug administration to the plasma, is the distribution, site of action, storage of tissue. Maybe the drug goes to the brain, to the excretion, free drug, protein bound drug. Again, here, this is the whole story of the systemic pharmacology. The biotransformation may occurs, and you can see, okay, remember these points. Biotransformation, either they can convert the active drug to inactive metabolites. You can see ibuprofen, paracetamol, chloroquine, or active drug to active byproducts. Inactive metabolites, active drug, phenacetin to acetoaminophen, morphine to morphine 6 glucuronide, digoxin to digitoxin to digoxin. Again, this is the active, this is the prodrugs, you can say. The active products. Inactive drug to active enhanced activity products like levodopa to carbidopa, prednisone to prednisolone. So this is the prodrug. These are the example of prodrugs. Some drugs that are not stable, but whenever they are converted, metabolized, they can convert it into active metabolite. Carbidopa is the active form. Prednisolone is the active form of the prednisone. Levodopa is the antihypertensive drug. This is the steroids. They can be converted to the prodrugs. And drug to reactive or harmful metabolite. So biotransformation also like isoniazid to acetyl isoniazid, and they have a very long, very high toxicities maybe appear. And you can see from here, isoenzymes, these are the substrates. Chlorine, there are various drugs, and these are the inducers and these are the inhibitors. Right, just remember this you can guys just can have a look over this. I have added some cytochrome P450 systems, their substrates, their inducers and their inhibitors. You got the differences between the inhibitor and the inducers, right? I'm not going to discuss it in more details. Okay, that was the story about all those phase 1 reactions that are most dependent on P450 systems. There are certain kind of phase 1 reactions that are not involving the P450 systems. What are that? Amine oxidation, for example, catecholamines, like oxidation of the catecholamine are histamines. They are mostly they are not involving the P450 systems. Right, alcohol dehydrogenation, for example, ethanol oxidation, steroids, for example, metabolism, prabastine in a liver, and hydrolysis, for example, proteins. So they are not utilizing, they are not catalyzing the P450 systems. They are utilizing certain other enzymes, as I mentioned in my previous slides. Okay, the phase 2 reactions. The phase 2 reactions mostly they're conjugation reactions, and if the metabolite from phase 1 metabolite is sufficiently polar, it can be excreted by the kidneys, as I mentioned in my previous topics. And the phase 1 metabolites are too lipophilic. It can be retained in the kidney tubules. So that's why the phase 2 reactions attached their endogenous substrates conjugation, such as glucuronic acid, sulfuric acid. These can be attached and make it more water soluble agents. This is, I just mentioned, another exception is 6-morphine 6 glucuronide. Remember that this is an exception case. Sometimes glucuronic acid attached to certain agents, they make it more water soluble. But here in this case, morphine, whenever this attached to glucuronic acid is the which it become more potent. This morphine 6 glucuronide. Remember this is more potent than morphine, right? So is the most glucuronic acid is the most common and the most important conjugation reactions. And if as I mentioned earlier, the conjugates, they have deficiency of this conjugating systems, they are more vulnerable to drug toxicities, especially against the chloramphenicol. I think baby fever, gray baby fever. I just forgot the name. That is the side effect of the chloramphenicol, especially in those neonates that have deficiency of the glucose phosphate, the glucuronic acid conjugation deficiencies. You can see the drug already possessing. Actually, remember this point, as I mentioned already, that they can directly enter into the phase 2 reaction directly. If there are certain substances that have this functional group already have that, so they can enter directly into phase 2 reactions. So that's all about the drug metabolisms. If you have any question, you can drop that in our chat group, chatting group.